Dear ladies and gentlemen, a warm welcome to our international webinar on navigating career paths in bioinformatics, from exploration to expertise. Today is a unique opportunity for each of you to unlock the treasure in the dynamic realm of bioinformatics. We are honored to have with us an exceptionally talented expert in the field, Ms. Ajita. She has a amassed 10 years of extensive experience in NGS pre-post sequencing, demonstrating proficiency in platforms such as ION ApplySec, ION Reporter, and clinical interpretation of NGS data. In June 2012, she achieved a notable accomplishment by securing the 8th nationwide rank in the Bioinformatics National Certification Exam. Throughout her career, she has authored 8 impactful publications that have significantly contributed valuable insights to the field of bioinformatics, showcasing her dedication to advancing knowledge and pushing the boundaries of scientific understanding. So, with her treasure, with the treasure of her knowledge, we are confident that by the end of this session, you will fulfill with the valuable knowledge that will stay with you always. So, without further delay, let me welcome Ms. Ajita. Hello, ma'am.
Hi, thanks for the nice welcome. Thank you.
Yeah, ma'am, we can start the session now.
We'll get started. Okay, sure.
Before starting the session, there is a notice for everyone that 100% attendance is mandatory for your certifications, and your attendance sheet will be provided at the end of the session in the 15 minutes.
Okay. So, yeah, I think the tone of the presentation was set well. So today we will look into navigating the career paths in bioinformatics. So thanks, Dr. Omex Labs, for this wonderful opportunity. It's a pleasure to be connecting with you all, a budding generation of bioinformaticians. So, yeah, do we have more people joining us, or should we just get started?
We can get started, ma'am.
Can get started. Okay. So the first thing I will do a quick exercise. So if you guys can, if you guys have um, a browser handy, you can just. I'm just gonna do this. Screen. Yes. Can you see my screen? Yes. Okay. So if you can join at menti.com, you can just type menti.com, and you can use this code there, 13500302. We'll just do a small exercise to get to know each other, and we'll start the session. Awesome. I can see some people joining. Great. Great. Okay. I can see thumbs ups. Great. Okay, so I would first try to understand what your core skills are, what's your background like. So this is an exercise for me also to understand that. So there'll be a box on your screen where you can just type what's your core skill. It could be like, biology or statistics or software engineering. So you can probably just type it in. So it'll show up here, and we'll come to know. Yeah. Somebody is texting that biotechnology. Okay, thank you. It'll be great if you can type it on the menti.com screen. Okay, great. Hmm. So there's a lot of biotechnologists. I see a lot of bioinformaticians also. That's wonderful. Yes.
Aditya, I would like to interrupt you here.
Yeah.
I'm making you host of this event,
Okay.
and
Sure.
in the security tab,
Uh-huh.
please allow the participant to unmute themselves or mute them according to your convenience. Is it?
Okay. Yeah.
You're the host now, and probably it would be fine if you can mute everybody.
Okay.
In the security tab, this option is
Got
available.
it. Got it. Yeah. Unmute themselves. Okay. So I muted everybody.
No, I'm not muted.
No. Okay. Hold on. Just a minute. I allowed. There's a option to say that they cannot unmute themselves. I've done that.
Yes, that was the option only.
Okay. I don't know how to turn particular people on to unmute, but yeah, just let me know if you want to, say something, and I'll switch it on for you guys. Okay. Okay, this is great. We have 46 responses so far, and this is wonderful. We have a lot of biotechnologists, a lot of bioinformaticians, structural bioinformatics, great biostatistics. Okay, data analysis. Okay, great. This is a wonderful group. Thank you. Okay, this is. Oops, sorry. Yeah, great. Okay, so thank you very much. This is a good starting point. I'll tell you about my background in a minute, but this is good to know. So we have a lot of variety, and we have a lot of people who have an understanding of the domain, which is a good starting point, and we can always build on the other skills. So great, thank you guys. Then we have another question where there are some options like, what are you most familiar with? Is it genomics, proteomics, drug discovery, cell biology, or math, or programming, or algorithms development? You can just pick one. Drug discovery is okay, great, very good. Yes, awesome. Really nice to see cell biology getting picked up. It is indeed the most fundamental thing that everything is built on. Awesome. Thank you. Thank you, Santosh. Great. Okay, this is fantastic. So I'm also happy to see that some people are interested or think they are most familiar with programming and genomics. this is wonderful. um, Bioinformatics is a field where all of us, everybody who knows a bit of biology or math or programming, we have everybody has a space and a good seat on the table. So this gives a good understanding of the audience for us and for you guys also to know the variety of people that are coming to such meetups, right? Okay. Great. Thank you everybody for participating in the exercise. Now let me just deep dive into the presentation. Okay. So I'm also from a similar background of you, like all of you. Um, I have been doing bioinformatics since 2012, so it's been close to 12 years, 11, 12 years now. And uh, initially I started working, so I did my bachelor's in bioinformatics, BTech in bioinformatics, and then I did a postgraduate program in artificial intelligence and machine learning. So that's my educational background, and since 2012 I have been working with genomics of rare disorders for a while, and then I moved on to software development, particularly with so genomics of rare diseases. I did it in TCS R&D, and then I moved on to Thermo Fisher for doing software development for genome sequencing instruments, ion torrent, and ion implicit panels. So was doing that, and I also explored cancer genomics there. And now currently I'm in the pharma, doing something for data management. I'll give you guys some tips on how to plan your transitions and how you can capitalize on what you're good at. Um, so roughly, I have a background that is a bit of bioinformatics, a bit of software engineering, and a bit of research data management. So the only reason I'm trying to tell you about my story is to for you guys to kind of build the confidence that there is some experience that I can help you with, and yeah, hopefully you'll pick up something that's interesting for you. um yeah, so let's get started. So first, let's get a little bit of introduction to bioinformatics and data science and life sciences. Some of you are, I did notice that some of you already have experience in this, but just in the interest of everybody, we'll briefly touch upon what the landscape is. So data science and bioinformatics is a very inherently related field. Even before the concept of data science became really familiar and a popular trend, we were doing data science in some other names, in the name of statistics, in the name of data mining. So bioinformatics is the core that is a combined intersection of computer science, math and statistics, and biology. So when you write software algorithms, you're doing something in the computer science world, and if you're writing it for a particular biological question or answering a biological procedure, then you're right, you're kind of into the bioinformatic realm. And there are also a lot of traditional research that goes on in different parts of the world. It's still what drives a lot of innovation and new learnings. We will be looking at one such example in today's presentation. So we do a lot of this for the um, CRISPR, for one concept, right? It involves it is a traditional research concept. So so there are a lot of research that happens there. And then when you actually integrate data mining or AI into it, and when you really mine insights or patterns from whatever answers, like if you write an algorithm to populate a particular data, and then actually deriving the insight from the data is a bioinformatician's job, right? So we are in the intersection of wonderful many fields. And there is medicine, and there is also translational medicine. So translation medicine is how you can translate the learnings from research into or experiments into actual medical diagnosis or treatments. So bioinformatics is actually interfacing with a lot of this. It could also be like, one of the translational medicine um, examples is not, there are also plants that are actually affected a lot by by new pests, and different farmers face a lot of problems like that. So actually developing methods or practices or solutions to address in a genetical modification or genetic engineering, or all of that could also come into this picture of bioinformatics. We will be looking into each of those individual items. So this is roughly about the data science and bioinformatics landscape. Now, what do we study? We study genes, we study genomes, we study proteins and metabolites. So these are the fundamental blocks. We also study one level up, which is the protein families. And then we also study the molecular structures. Then we study chemical compounds. This could be in the body, outside the body, in vivo, in vitro, all of that. This is basically for the chem informatics aspects of things. And then there is, so these are things that we mainly study. And how do we study is using different concepts. We use systems biology, and we also use lines of study like immunology, or such things also come inside. And then we take the help of algorithms and software development to enable us to do do this research. And eventually the central crux of this is, this is my perspective. There could be other things as well, but from my perspective, this could be, you could have disease management, pharmaceutical industry, and digital sciences. So these three are the main pillars that we usually revolve around. So from exploring evolution of human genetics to measuring efficacy of drug response, bioinformatics impacts many areas of medicine. So that's like, the whole, it's a completely big spectrum that we cover. Now, just to understand how far we have come and how promising the future is for us, some of the highlights from the history of bioinformatics. My personal favorite is the Human Genome Project. A lot of bioinformatics wouldn't have been possible without the Human Genome Project. There was Greg Venter and very many institutions, including industries and academia. Everybody came together for this project, and there were many countries that were involved throughout the world, and we all participated in this Human Genome Project, and that is when we get a consensus human genome. Like a fully annotated, like we understood all of what the human genome has and what are the individual bases. So all of the sequencing, that was the first time we saw like the complete picture of human genome. There are still some more iterations going on to to clearly understand telomeres, or to make, like, the initial days there was just one genome, like a single assembly, and now they are having alternate context to say that, okay, this is a part of the genome, it's supposed to have ATGC GT, and this is the sequence that's supposed to have. But there is also an alternate context that's very prevalent. So this region could look like this in like 40% of the population in the world. So we are trying to get such alternate contexts and more clearer definitions for telomeres. And so it's still growing, but the first was happening from 1990 to 2003, and I think from then the boom of the bioinformatics really kick-started, and we have just been on the uphill. And then we have the 1000 Genome Project. This is also a wonderful project. If you guys know about minor allele frequencies, which is one of the key parameters in any of the NGS analysis that you do, you will definitely have come across the 1000 genome project. It tells you about, in a chromosomal position, what is the most common allele and what is the second most common allele. It just gives you those details. So that is a repertoire of information that is like really unparalleled. And then there are some questions. Okay. So then we have the advent of all sequencing machines and software. One of the key objectives of big companies like Illumina, Thermo Fisher, and PacBio, Nanopore, all of this is one of the key objectives they have is democratizing the sequencing, right? How cheap can you go, and how fast can you go, right? So that is making a lot of things possible. There was even there was a small like a pen drive. You can sequence your DNA with just an instrument that's the size of a pen drive, and that was taken to space, and it was they used to sequence it there, to sequence something there. So it's just like it opens up a lot of possibilities because of the availability of sequencing machines and software we have. And then my next favorite is the encode or the encode project. So this was probably in 2013 or 2014. I was there in TCS Hyderabad, and my boss came and he said, did you see Nature publications? There is about like 10-15 papers that was published on overnight, that said they had understood how 80% of the human DNA, what is it for. And they said, we now know what 80% of human DNA does. That was a huge declaration, because exomes, or the gene regions of the genome, are not too much. Like, it's like, usually the exomes that we sequence are like 3%, or maximum 1 or 2 to 3% is what we usually sequence for understanding diseases. So somebody telling us that 80% of the human DNA, there's a lot of introns, there's a lot of junk DNA. They just said they know everything. So it was like a huge milestone in the bioinformatics world. And that and they mentioned they understood some regions are there because they are going to bind transcription factors, they're going to be DNA methylation sites, epigenetics. They had a lot of explanations to a lot of DNA that we didn't know before. So but it was a very big controversy at that time. People were like, well, I don't think 80% of the human DNA is needed. It's just that there's this is junk DNA. Then people were then the onion theory came into picture. They said like an onion has 5 times more DNA than human. We are a very complex organism. We have a lot of biological processes, but why will an onion have a lot of DNA than us, right? So maybe there are some DNA that is indeed junk. It's just that it's really not evolutionary impacted yet, but there could be a function to it also. So there are a lot of questions still that we can still answers. And understanding non-coding DNA is a big research area right now. So this project opened up another big avenue for us to explore. And then we got the attention of big companies like Google and Microsoft. And Google came up with a variant calling software called Deep Variant. And so when they came in, they were uh, sorry when they came in, they had actually taken it to the next level. They started using deep neural networks or artificial intelligence, and they were doing genomics. So artificial intelligence and genomics, they again opened up. They were like leading the field with new things like that. And since then, there is no looking back. There's a lot of genomic data repositories we have. So a lot of data means a lot of ways to analyze genomes, a lot of insights to be drawn, a lot of research questions that could be answered. So it just kept growing. So this is just a small example of what has been happening in the past 10-15 years, but there is a lot more. And it's still happening. So we have a very promising future. So yeah. And moving on to some of the concepts of bioinformatics and data science. So what is next generation sequencing? This is one of the key areas of bioinformatics that has the highest market share, the highest job opportunities, and everything. So what is this thing? So next generation sequencing is nothing but making copies of the genome to understand the genetic variability. You just take one DNA, one DNA read, or one copy of the DNA, amplify it a lot more, so that you can understand what is the exact uh, base in that position, what is the exact nucleotide in that position. And with this, you can know if the patient has a mutation or not. And depending on if they have a mutation or not, we can suggest them what medications or treatments that they have. So first, a patient, when he sees that he has some problem that is not a conventional problem, they go to the doctor, and the doctor usually asks them for their clinical phenotypes. Phenotypes are the observed changes, right? They look at the phenotypes, they look at what environment they are in, and then they also do a lot of molecular analysis. And with this in the picture, they prescribed some medicines, or they designed a treatment plan. So this whole section here, that's molecular analysis, this all of this comes into lipidomics. And lipidomics is not really into NGS yet, but there are a lot of epigenomics, genomics, metabolomics, proteomics, transcriptomics. All of this can come into in the NGS realm. Then and if you ask how these are individually related, so first we start with phenomics, which is the what we see in a person. Like, there is I'll tell you some stories in a couple of slides, but there is what do you see in the person? Is he having difficulty breathing, or compromised immune system, or diabetes, or some cancerous traits, or this is what they call as phenomics, the morphological, behavioral, and development patterns. And then comes the genomics. Genomics is a very famous, and a very, it has a long history. So this is a study of sequence level DNA modifications. And then we so there is a DNA, and there is the epigenomic layer that wraps the DNA, like for people who don't know about epigenetics, if you take a DNA in one cell of a person, and if you stretch it, it'll probably, the double strand, if you just stretch it, it might probably be like 2 meter long. So how it is all compressed into like a really micrometer, or a very very small cell, right? So this condensation happens in the epigenome, or the chromatin. So there are proteins above which the DNA is rolled, and this DNA comes out for transcription. So this network is a very key. This chromatin plays a very important role. If there is a gene that is that's in the DNA, that's wound on a chromatin, but now when it's not coming out, so this gene goes unexpressed, or there is a down regulation of this gene. So this epigenetic modifier also plays a role in how the genes are transcribed or not transcribed. So the study of non-sequence level. So there is a this is not on a DNA, but the chromatin has the problem. So it is the problem in the chromatin is not letting the DNA to come out. So the study of such chromatin or such epigenetics is also a huge area of interest. And then you have transcriptomics. After the DNA is transcribed, what happens, and then when it is translated into a protein, what happens, and then the eventual metabolites, how they are all related. So this is how the story unfolds. Bioinformaticians come in at different points, and they perform. So we are in capturing the information as a genetic counselor. We are in the genomics phase as doing the actual analysis, building the tools for the analysis, performing the analysis, communicating the analysis results to the stakeholders. So we come in at different levels on all these lines. Now, then about the diversity of an NGS portfolio is that you can study the entire genome, or you can study only the exome, which is like the most meaningful region of the genome, or you can just study the clinical exome. This is a buzzword that some of you might have heard. Clinical exome is the actionable exome. Like I can study the whole, all of the genes, but only some genes have a medicine or a pharmacokinetic or a pharmacodynamic interpretation associated with it. So then if I study only these in these regions that are actionable, then you call it the clinical exome. And then you have targeted panels, which are like really like, I know breast cancer is caused by two mutations. So, in this type of inherited breast cancer types in this population, it's usually these two mutations. So let me just sample for them first. So you're creating a very narrow panel which is like very focused. So then you can use targeted panel. So how you do the portfolio, how do you do the NGS analysis also has an interesting portfolio. And then you can there are different applications on how you can do it. You can do microbial resequencing, you can do eukaryotic resequencing, you can do de novo, shotgun plasmids, you can sequence many things in many different ways. And you can study diseases, you can study you can study metagenomes, you can study populations, forensics. We'll get into these one by one. So when you're doing all this, so when I'm looking at exome or whole genome, or what I eventually want to know, what is the end result in my mind, is if I can find biomarkers. Can I tell that this person has this mutation which is causing a disease? That's what eventually I want to get into. So one concept that you will usually hear in this world is biomarkers. It could be single nucleotide variations, deletions, inversions, translocations, all types of variants. And this is how a typical NGS result will look like. You will have different reads. So if you see there is a cytosine that is seen in most of the places. So it will just say that, okay, it's supposed to have a T, but I'm seeing a C, and I'm sure it is a mutation because a lot of reads have the C. So so that is a confidence. And that's how the process goes. And yeah, and sometimes if it is not a mutation, if you want to look at structural variants like copy numbers, you'll know that, okay, there is a chromosome 4, there is a particular region of the chromosome that is, it's not having a double ploidy, it's having a single ploidy. So like it's like a karyotyping, right? Like the, you can imagine like a very accurate deep karyotyping of chromosomes. So we can do all of that. Now this is what we are eventually going to look at. And we have uh, and almost everything that you do in bioinformatics and NGS will revolve around something about genetic diseases and inheritance models. There are a lot of inheritance models. I'll just show you some of the I these are not conventional. You would have heard about dominant, recessive, yeah, compound heterozygous and all that. So, and autosomal recessive is one where this is also probably common, where you just have it's a recessive one in the parent, both the parents, and when both are inherited by the child, it becomes he becomes affected. But X-linked dominant, I don't know if you have heard about this inheritance model, but it's only in the X chromosome, and the father is affected because he has only one copy, and that copy has a problem. And if it is a dominant, the mother has a normal X chromosome, so she doesn't have a problem. But still, when the daughter inherits it, then she becomes she has the phenotype, or she has the disease because she has the dominant allele, and she inherited it. So um, yeah, so these are some of the new models. You can read about them in the NIH link. There a lot of new models are being described as well. Yeah, you can just read through them. So these are all very important information that will eventually build our pipelines on. So let's go to a story of how this was done. Um, so this is a work we did long back. It's an interesting story. There was a child. So in the US, this is also coming in India, but in US it's very famous. It's called the newborn screening. Whenever a child is born, they'll have a list of diseases they screen the child for, and if they don't have an explanation, they'll send the genome of the child to somebody to analyze and tell them what it is. So one such samples came to us, and that patient is a small baby, and he had a lot of bacterial infections, and and he also had. So first when he comes, we ask about phenotypes, right? So we asked what is it, and they said immune function is a little bit of immune function is preserved, but his immune system is otherwise highly compromised. He has a lot of infections. He also has craniofacial and dermal abnormalities, and he has an absence of corpus callosum. So they gave us some inputs like this, and then what we did was we sequenced his genome. We got about 5,000 protein altering variants. So when I mean protein altering variants, we are studying the exome here. So we are studying the genes, and we are trying to see is there. So can someone tell me what types of mutations will be protein altering? Like for example, you will have synonymous mutations, non-synonymous mutations, missense mutations, nonsense mutations, frame shift. So there are a lot of mutation types, right? So if you guys know something, you can type it in the chat. Yeah. So there are a lot of protein altering variants, and can you imagine that there are like a billion base pairs, and what we see is just 5,000 protein altering variants. So when you actually have a good focus of what you're looking at, you will be able to get to the biomarker easily. And then we wanted to do a quality check on this. Somebody is saying that non-synonymous and frame shift in not multiple of 3. Yes, good. That is a that's very good. Yeah. So deletions, additions, transversions. Yes, that's also a good thing. Yeah, correct. Um, so yeah, so there are so first we did a protein altering check, because any synonymous mutation, even though it's a mutation, it's not going to have an effect. So let's keep that aside and only look at protein altering variants. And then we did a quality check, and they said there are 3,000. So this quality check is about the sequencing quality, like how confident are you that there is a mutation here. So when we did all that, we got about 3,300 variants. And then this is where the 1,000 genomes helped us. So we know that this disease is a very rare disease. It's not like like a cardiovascular abnormality or something. It's not a common one. It's a highly rare disease. So when we look at such diseases, we went into the 1,000 genomes. So they actually, they're like 1,000 plus. It's not actually exactly 1,000. There were more than it. So there were like 1,000 genomes plus were identified. And if you find that there is a variant that's present in more than 2% or more than 5% of the population, it's not a rare variant anymore, right? Because it's still, even in 1,000 people, if you can find it in 2 people, it's still a common mutation, we felt. And we put a threshold of 0.2. These are all empirical values. You can keep changing them. So when we did that, we found 866 rare variants. And then finally we put the inheritance models which we saw previously, and then we found 57. Now we go into gene prioritization to see which genes are important. And then we noticed that three genes had five variants. Then this goes into the eventual experimental testing. So we tested them in zebrafish, and eventually the BCL11B was chosen, and accordingly treatments were given. So this kind of tells us, like, there was a baby with a disease. He had a treatment. In between, there are multiple points where bioinformaticians came in, and we kind of solved the problem for them. So and these are, you could imagine how impactful this exercise would have been, right? You are actually suggesting a child that this is the causative mutation, and they treat it, and the child was actually, he got back his immune function. It was restored to a good extent. So um this is how bioinformatics truly make a difference. And then you have relatively common disorders, like diabetes. So even with diabetes, it makes we can do a lot of different. It's not just rare diseases, right? So there are about 552 million cases by 220. It is projected by 2030. This is a lot of people. And you could you know there is a famous doctor, Dr. V Mohan, in Chennai, and he's doing a lot of efforts. He's putting a lot of efforts into these, because there is diabetes 1, diabetes 2. There are different types of diabetes also, right? So um, the treatments have to be different for each of them. And that is one, he actually told us about a story. Dr. Mohan was telling us. So there was a girl. She was actually given insulin on a daily basis. Her diabetes was so bad. But then he did a genetic testing, and he realized that she doesn't have she doesn't have a particular a particular gene was not doing what it was supposed to. There was down regulation there. So they gave her a different medication, and then she was not insulin dependent anymore. So there were I don't remember the exact details, but these stories are very famous. And so why should we give them insulin when they technically don't need it, right? It's because of ignorance that we were providing them such treatment. So precision medicine is very important, even for very common diseases like diabetes. So this is an example of how many markers we have identified for diabetes. And eventually what you want to do is you want to create a plan in and these plans are also formulated by bioinformaticians. So this plan is given to a clinic. So there's a genetic counselor who sits when a patient comes. You can tell them, okay, do you have if the patient is with the thalassemia, right? Do you do any genetic analysis? Was there any alpha globin gene deletion? Then okay, then this is what we should be doing. If there is no deletions or something, then this is what we should be doing. So there's a clear cut plan of what needs to be done at depending on the genetic makeup of the patient. So such plans are very important for us to decide the right treatment plan. And all of this knowledge is built by bioinformaticians like us, using different tools and data. So yeah. And with genomic data science, we have identified a lot of diseases, a lot of causative genes for diseases. So we are making people understand the disease more. So once you understand it well, you can treat it also well, right? So we are helping people understand very nicely. And this is a data that was generated mid of 2020, and that's why you see a small bar, but it's actually growing still. So the trend of the curve is very real. It just keeps going. And yeah. um okay, so maybe we'll just pause for a minute here and ask for questions. I'm letting people unmute themselves for so before we go into NGS of oncology, which is another new realm. Yeah, does anybody have any questions? You should be able to unmute. If not, just let me know. Anybody? I hope it's clear. Okay, just put it on the screen. If you just type in the chat if you have any questions, and then we'll. Is Trinity good for RNA-seq? I got a direct message. How NGS role play in CRISPR's technology? I have not been doing RNA-seq in the recent times, so I'm sorry, I'm not sure about the Trinity question. NGS role in CRISPR. So CRISPR is actually um, so NGS and CRISPR are related in a few ways. NGS is like the high level exploration, and CRISPR is like you really know what you want to um, target, and you know what you want to study about, right? Exact gene, and then you can put a plasmid and correct CRISPR tags, and then you can attack that particular gene and see how that responds. So one thing I've seen is people like in this uh, like in this exercise. So what we do is, at this point we tell them that computationally we have checked and there are 5 variants in 3 genes, so these 5 variants are my suggestion. So then what they do next is they actually test these 5 genes in a mouse model or a zebrafish model, and they see whether the gene, if you if you have this mutation, is a gene silenced or not? That's the question that they ask. For those validation purposes, CRISPR also is a very important, is a very important tool. So yeah, I think that's where NGS and CRISPR relate to each other. I'm not sure if there could be other avenues, but yeah, this is what I'm thinking. um what are the ways to treat diabetes without insulin? Yeah, there are a lot of ways to treat. See, if the diabetes, it's about the what is the molecular basis of the diabetes, right? If the patient does not have an inherent capability to produce insulin at all, that's when you have to give them insulin. And even it's like if there are any metabolites that's not available to to digest some products and make it insulin, then you can just provide interfere at that point and make sure those metabolites are produced, the insulin production goes on. So where is the insulin production getting disturbed? Is it from the receptors or metabolites, or there's a you need to study the system, and that would also be explained by the genetic makeup. If there are some receptor, where is the mutation in this pathway? So you can point to that, and you can see. So diabetes can be treated without insulin in many ways. They can just control the glucose level, and there are a lot of ways to do that. um do you anticipate the DNA sequencing cost to go anytime soon? Cost has not been going down since last 5 years. I'm not very sure about that, because I was in Thermo Fisher till 2022, and I did see some of the cost going down. Well, if the it's like, what are you studying? If you're studying the whole exome, do you still need to study the whole exome? Can you study a narrow region? And the moment you are reducing that, your cost automatically goes down. And and there are also even Illumina had a newer system which was still cheap. I think the you know there are certain aspects to it. How costly is the instrument? How costly is the panel? How costly is your reagents? And how costly is it to actually maintain it? So which one, we'll have to see where the cost comes from. But the cost of the machines are really going down. The cost of the panels are also going down. So that's one thing. Where do we have best and affordable means to get whole genome and RNA sequence in India? Oh, there are a lot of institutions. Um, mm, there are a lot of, I don't know if I can name them here, but I have my colleagues in or my friends in Strand Acid Medgenomes, Genotypic and Doctromics. There are a lot of institutions that do it. Yeah, okay, cool. Is it possible to sequence specific genes using freeware and online to, well, you cannot sequence without running the sample, right? So you'll need to take a sample in a wet lab, and you will have to sequence that to see what is the exact mutation, right? So that, the sample, basically a sample, by sample I mean a blood sample or a buccal swab or something. So yeah, that part has to be done in the wet lab. Okay. All right, now let's Bioinformatics with the no basics. Okay, cool. We will get into some of the. Let's go further, and then you guys can continue asking your questions. Okay, the next is about oncology. This is a very interesting field. Anything you do in oncology is a discovery, because it's such a complex field, and it's a very intelligent disease. So we need all guns blazing to tackle this. So yeah. So why do we need clinical sequencing in oncology? Is that when you do a conventional approach and when you treat, some people have benefits, some people do not have benefit, and they also have adverse effects. So the this is a survey that was done. I've given the link, and they mentioned there's a 30% clinical response in the population that is in the sample that they studied. But if you do clinical sequencing, and if you know, if you are if you have an understanding of who is going to respond and who is not going to respond, so first you can you can reduce the strain on the patient by not giving him the wrong treatment, and second, you'll have a good response depending on their genetic makeup. So there was a group which got 75% respond. There's a group which got a different treatment because of their genetic makeup, and they got 67% respond, and another also 67%. So you can see that depending on the genetic makeup, we really need to shift our gears. And the key point here is bioinformatics should and is focusing to improve response and decreasing the adverse effects. Now, Dr. James Allison and Professor Tosuku, he's very famous. I'm not sure. I'm sure many of you know them. So they got a recent Nobel Prize, and what they did note, what they gave to the world was the concept of immune therapies. Immune therapies have been there for a long time, but they actually made substantial improvements on how it can be used for cancer. So why do you need immune therapy? If for people who don't know, immune therapy is in traditional treatments, there is either a drug or a radiation that's given, and they just they just target towards the entire region, the cancerous region. It could have healthy cells, it could have cancerous cells. So there is a full-blown effect of that. But if you think about cancer immunotherapies, first what we do is we look at the immune repertoire of the patient. By this, what we mean is we look at the B cells, the T cells repertoire, or what's the genetic sequence of the B cells, the large the chains in the immune globulins, immunoglobulins. What are the chains, IgIs and IgBs, and what is it actually having, and why is this not able to tackle the cancer cell? Why is it not understanding that the cancer cell is a foreign cell? So if we tell them that, if you have this chain, then this could look at the cancer cell, and it can understand the cancer cell as a foreign cell, and it can attack it. So this the understanding of why a patient's immune system is not recognizing a cancer cell as a foreign cell, and helping them to have that extra immune cell to attack their own immune system, is cancer immunotherapy. So there is very less side effects, and there is a really good post treatment protection as well. And this is also a very high impact NGS activity, where we look at the repertoire and we tell them which works and which doesn't. So there are NGS panels for this, and so we are playing a huge role in cancer immunotherapy. And we also study a system, not just one cell's DNA, or one particular immune repertoire is also, we are studying like a repertoire, no, so like a system of immune cells. So we also study tumor microenvironment, what are all the metabolites and everything that are available. We look at we do evolutionary studies, we do single cell RNA. So this, like a cell snapshots of what is in the cell at one point of time. We do that as well. And yeah, so like on a time based, like what is the progression of things over a period of time, what is the changes in the morphology, or this the abundance of a particular transcript over a period of time. So we do things like that as well. And spatial omics is also something that's really catching up. So we study systems in general. And how do we do that is, we there are very three main levels to understanding systems. One is we try to mathematically model the systems, and then we understand the information flow, and then we see like, okay, now if you look at this network, there are genes, they are connected, there are information that flows to them, and then there are like sinks, and this is how interaction happens. Now when I remove this, when this gene is silenced, what happens to the rest of the network? So you can mathematically model all this. This is what systems biology is about. So a lot of bioinformatics happens on this side as well. And this needs math, understanding of math, a little bit of physics if you're interested in. So biophysics is also related to this. People do flux balance analysis, assuming this is like a electrical flow system, like how much of like the information that goes in and that goes out at the time, they try to balance that. So there are a lot of interesting concepts and allied fields that come here. Yeah, and I personally did an experiment when we studied like if one gene is silenced, what happens to the other system. And we created some interpretations of how apoptosis is silenced, and that is very important for cancer to be attacked, to be treatment plans to be done for that, right? So these are some things that we can do. And keeping aside the humans, we can also study microbes. So this is an example of a dairy sample. So uh, there was there was a dairy product. I think it is a milk factory. So they were just taking samples of milk, and they were trying to see what is the species that are available, that are present in this particular sample. So for this, first you need to have a reference database of all the possible species. And then you would have heard about blast, right? So you take blast, and then you do NGS. You take the sequences of the current in the current sample using NGS. You blast the sequence in your sample with the reference database that you built, and then you we do multiple sequence alignments to understand which are actually present here. So there are phylogeny uh, that is done on top of this. So it's a very interesting field. So there's NGS, there is a regular usage of tools like caste and species, and so metagenomics can also be understood this way. And we also studied viruses when tackle the variants. So people are understanding what variants of COVID are going around, and which is the most pathogenic variant. And there were a lot of discussions around that to understand how it's evolving and what's happening. So here IGIB was very very pivotal in doing this activity across, and there were a lot of sequencing efforts going around. So who was doing all this was institutions in India, startups, and many some larger institutions in India. They were getting samples and doing all this. There were bioinformaticians sitting and collating the information. So yeah, so this is also a very recent activity, and since then there is an investment increase from the government of India, and the DST, DBT, and everybody is actually thinking of setting up proper investments for doing bioinformatics. So these are all good things that will increase job opportunities and everything for us. And um, this was mainly done by Vinod Sakaria, Dr. Sridhar Sivasavu. There are many stalwarts who are doing amazing jobs in the field of bioinformatics in India as well. And then we also have forensics. This is a very interesting field because in the US they are using it a lot for solving crimes. They actually take hair samples and any skin or anything that's available in the criminal scene. They just take it and they do all these analysis to see, and they run it over their criminals and crime database to see who could be the who are the people present on a site. They're doing a lot of this. So we use there are many things you can use, but STRs is highly catching up in the field of forensics. So people study how much replicates. STR is a short tandem repeats, right? So how many repeats are present and the combination of repeats could be different in different people, and that's a marker, right? Because it's unique for everybody. So at this time we should also you guys, if you're interested, I suggest you also learn about GDPR and genomic data reporting policies. So one ask by many institutions, for many institutions that are reporting genetic data is how well are you making your software GDPR compliant, like how much anonymity are you maintaining, so that the patient who is involved in the genetic and whose data is involved in the genetic analysis is protected. And how do you report your results, like for example, you can imagine a scenario where you're looking for a disease. You find some variants, and you're trying to understand what does variants mean, and you find that he's prone to a particular disease. This mutation likely causes a particular disease, but that patient did not come for the diagnosis of that disease. He came for something else. So now, but you do know that there's another disease he's prone to. So what do you tell them? Do you tell them that you came to us for a cardiovascular disease, but I think you are prone to have some other metabolic disease? You might get that. Do you tell them, or that's unnecessary stress for the patient? So how do you report these things? There are very clear guidelines on what you should do in those scenarios. So as people working with human genomes, this is something we should all try to understand. um, So that is one thing. And for fun, people also look at evolutionary plants and animals. This is also a very interesting field. These lichens, they actually took specimen that were 138 years old, and they still understood that 70%, 75% of the DNA could be fine sequenced very well. So it's a very powerful tool. Even dried blood samples that are, people give blood samples. They just there was once there was a lab where they had a bag full of dried blood samples. It was just a very dried one. Then they asked us, we have the fresh sample of the same person. Now can we compare what happened between this, like when he came 20 years ago and gave us the sample, how was his genetic makeup and now how it is, can we compare? So there are you still can do it even if it is a dried old blood. People still can recover sequences from there. So um, these can explain. And in terms of plants and historical things like that, we can actually understand how to preserve them, how old they are, and understand their characteristics. Yeah, so going forward, where is the field heading to? The field is heading to, I personally like this line of study, where we are not just trying to understand does he have a disease, but instead we are trying to understand how much disease is there in this person. How far is it, what is the stage, and things like that. We can get to this. So depending on so this is a particular example where they were trying to understand disease progress of Duchenne muscular dystrophy, the DMD one. So they actually came up with a nice chart of how how far it is, like is it late loss or late onset, or how, which genotypes are present at which stage. And with that, you can easily understand and place them in the correct categories and start treatments as expected.